So good morning, everyone. My name is Hákonarson from Children's Hospital of Philadelphia, and with me today is Dr. Huiqi Q, physician scientist, similarly from Children's Hospital of Philadelphia. And we are members and part of the IHCC team, Scientific Strategy and Cohort Enhancement. And Huiqi in particular has been focusing on polygenic risk scores, generating polygenic risk scores across multiple cohorts within the IHCC network. And he is going to present to us today some of his work, and mostly focusing on how we have been able, because of the nature of the IHCC, to leverage trans-ancestry approaches to assessing polygenic risk scores. And he will similarly tell us a little bit how you can also apply those techniques on the metabolomic data that we have generated, because, I mean, you can, of course, take a vitamin D deficiency or any fatty acid deficiency or metabolomic factor and use that as a trait and generate polygenic risk. So we're looking forward to this session, and following the presentation, we will have a discussion sessions to sort of try to put together what this means. So Huiqi, please go ahead and go through your presentation.
Thank you. Good morning, everybody. Welcome for this meeting. So I will present some technical details and share it with you and about our trans-ancestry approaches to assessing polygenic risk score for Alzheimer's disease. Also some technical details about our metabolomic project with IHCC. So the IHCC enable us to share research resources to promote scientific research. So among different resources we can share, for example, method results data. So to share method have actually the least obstacles. So today in our review about the trans-ancestry approaches for PRIs, we actually created the model in our in-house data site. And then we share the method with our collaborator across the world for assessing its validity and efficiency. Also, in our metabolomic study, we did the research in the minority population, and we are happy to share our approach. More importantly, we are happy to share the individual data of this minority population, that is African American. So first, I will introduce a little bit about our PRI's work for the prediction of Alzheimer's disease. So the goal of PRIs is to combine the effect size of multiple variants into a single score. In this way, we can predict the risk of a disease or a phenotype. So there are many important applications for PRIs. For example, we can identify those patients with high risk of a particular disease that can be prevented by early intervention. Also, we can use PRIs to identify those patients with higher risk and give these people more attention to enable early diagnosis of the disease. Otherwise, for some disease, it's too late if we wait to the clinical diagnosis of the disease. For example, type 1 diabetes, once it's diagnosed clinically, it's too late for any intervention to reverse the disease. Also, the PRIs enable us to identify new subtypes of a disease. For example, we can identify those patients with very low PRI score. This may suggest these patients have special types of the disease, and there may be particular gene targets we can use to develop new therapeutics for these rare disease types. Also, we can use PRIs to predict the therapeutic effects, that is to predict the probability of benefit of the treatment. So currently, the major challenge is to address the issue of health disparity related to the lack of genomic information in minority populations. We know that the majority GWAS and genomic study have been done in European population. So there are two interchangeable terms for GRIs or PRIs, that is genetic risk score or polygenic risk score. So, but usually we refer GRIs as a group of selected genetic markers. So this group tends to have smaller number of genetic markers, and each marker may have larger genetic effect sizes. And in contrast, PRIs tend to include a large number of genetic markers identified by genome-wide study, for example, GWAS or genomic sequencing. And also, this group of markers have been selected by GWAS study, but this selection was mainly based on a threshold of the p-value, not by the selection of their functional impacts. And because the large number of genetic markers, genetic marker with smaller effect sizes may be included in the PRI scoring system. So first, I will show you an excellent example of a genetic risk score that is for the prediction of type 1 diabetes developed by Sharp et al. And different from other complex diseases, the GRS of type 1 diabetes is very complicated because it involves the major genetic effects from the HLA locus. In the HLA locus, there are interactions between different HLA haplotypes. There are also effect sites from other HLA loci genes, and also a large number of loci from other GWAS regions. So, but for the most other complex diseases, we can only use a simple formula like this. We only need to sum up the effect size of all the GWAS loci. So it's much simpler than the GRS for type 1 diabetes. For example, if we have a disease, we selected 50 markers, so we just add up to the effect size of the 50 loci. For each locus, we use these two numbers to calculate the value. That is, we use a beta value. This also always produced by a genetic association study. Or if you can see a odds ratio, we can easily convert the odds ratio to beta value. This n represents the number of the risk value. For example, if it's a homozygous, so this n is 2. If it's a heterozygous, this n is 1. So if it's a homozygous of the protecting allele, then we have a 0 here. So for most complex diseases, the GRS is very simple with a simple formula. So for PRS, there are many excellent algorithms we can use. For example, there is the PRECISE2 program, and there is the LDPRED2 program. So there are some difference among different algorithms. For example, if we are using PRECISE2, we need to use individual GWAS data to train the model of the PRS scoring. But for the LDPRED, we can just use the summarized data for the statistical association and for the linkage disequilibrium value. And then we can train the model, and then we can share the data, share the model for validation in different populations. So we know that the health disparity has been recognized for many years. And actually, since the beginning of GWAS study, the majority have been done in European population. So in our experience, we just created the GRS model in a European population, and then we share our model with different collaborators across the world. That is different international cohort in the IHCC project. And then different cohort assess the performance of the GRS scoring system in different human populations. And then, if possible, we can improve the model by their validation in different cohorts. So again, I want to use the type 1 diabetes GRS as an example. So we can see, we know that the development of the GRS for type 1 diabetes has been done in European population. And if we assess this model in African American, we can see that African patients have much lower score if we compare with the European patients. Again, for the controls, for the healthy controls, and we also see much lower score than the European controls. But interesting, we can see that the African cases has much higher score than the healthy controls. So they suggest the applicable of the GRI system for the minority population. And then we assess the performance of the GRIs for type 1 diabetes. In European, we can see it has a good AOC score of 0.823. And it has a lower AOC score in African American. It's about 0.07. And in this case, we included 4 more SNP markers identified by a GWAS in African American population. So that is a study led by Dr. Rich. And then we can see that after including only 4 more SNPs, we can see the AOC score is much more improved in the African American population. So now it has a AOC score of 0.826. So we can see it's even higher, a little bit higher than the original European American population, which the GRI score has been created. So also we can see that the GRI score is further improved in the European American population. So we have this program available in the GitHub. So anyone want to try this algorithm or want to apply this in your clinical study, so you are welcome to download it from there. So for the IHCC project, we developed this GRS model in a large European cohort for Alzheimer's disease. So we call this as a GIRA model, that is genomic informed risk assessment model, because we not only included the PRS by genomic marker identified by GWAS studies, we also include a major genetic effect from the ApoE locus. Also, we include 2 more important risk marker factor for Alzheimer's disease, that is the age effect and the sex effect in the Alzheimer's disease. We know that male has much higher risk than female. So after we created the model in European population, we first assessed the genetic polymorphism frequencies in different populations using the eMERGE 1 to 3 data set. And then we assessed the frequency of the genetic marker in African population from the cohort led up by Professor Ramsey and in South Africa. And then we sent our model to our collaborators in different international cohorts, and they try to assess the model in their population. So we can see that for every cohort, the best performance or the largest AOC score is actually seen in the full GIRA model. So we can see that for the 2 African cohorts with different phenotypes of Alzheimer, we can see the AOC score is much higher than the AOC of the genetic score alone. So that is the GIRA model. So for the South Asian cohort in East London, we can also see much higher AOC score than the genetic score alone. So for the Japan cohort, we have better scores. For the UK London cohort, we also see better AOC score for the GIRA model. So this is our experience, that is we created the model in the European population, and then we share it with our collaborators to assess it in minority populations. So now it's the second project we have done with IHCC, that is we have done the metabolomics study in the minority population, and then we are happy to share the data with you for your study. So in this metabolomic study, we use the Nightingale NMR metaplatform, and this platform includes 249 metabolomic markers that have been selected by their clinical importance by Nightingale. And the important thing for this platform is they include a large number of biomarkers for different lipoprotein subclasses. So this has not been done by other metabolomic platform. And also it includes 70 biomarkers for relative concentrations of different lipoproteins. And then 18 biomarkers for fatty acids and 9 markers for phospholipids and other lipids. So this is the way we want to bring your attention to about how to share our data so we can compare our data from different studies. So we can see that there is this panel include the 98 biomarkers for lipoprotein subclasses. So in our presentation of the results, we rank the biomarkers by their size. That is, the upmost is the actual large lipoprotein markers, that is cholesterol, cholesterol ester, and others. And then we rank the markers by their density. With their density increase, so we rank this until the very last. The highest density is those biomarkers in high density lipoproteins. So if we use the similar presentation template, we can compare easily our results. So this is our study of the sickle cell disease results. We identify a large number of lipoprotein, and so this is the 70 markers for the relative lipoprotein lipid concentrations. Again, we rank the markers by their density and by their size of their particles. So this panel shows all the left biomarkers included in the metabolomic platform. So that includes all the other groups, the amino acid and apolipoprotein and triglyceride and others. So we show these results by the forest plot, so it can be compared easily across different studies. So for the data analysis, we normalize the biomarker levels by means and standard deviation. This is how we calculate how we do the normalization. So by the average value and by the standard deviation. And then we tested the correlation between the biomarkers and the disease status for asthma, for sickle cell disease. And importantly, we control for the risk factor for each disease. For example, the age effects and sex effects are both risk factor for sickle cell disease that has been corrected in our correlation study. So we use the 0.0 partial correlations for this correlation test. So this is our sickle cell disease study. We identify a large number of significant biomarkers. For example, for the lipoprotein subclasses, we identify 41 markers are statistically significant. And that means 41% of all the markers are significant. So when we are talking about the significant, all the significant eye level has been corrected by the bifurcating correction for multiple testings. So these are corrected p-value we have used for the significant results. Also, for many other markers, we've seen lots of significant markers. So this is the conclusion we can acquire by our sickle cell study. So we identify hypolipoproteinemia and hypocholesterolemia in lipoprotein subclasses, but these are only seen in those subclasses other than the very large density lipoproteins. And also we identify decreased plasma free fatty acids, including the total fatty acid and saturated fatty acid and unsaturated fatty acid. So these results may suggest dietary supplementation of the deficient nutrients may be beneficial for the disease. But we also want to bring people's attention that some decreased nutrients or biomarkers may be the result of the disease. It may have a detrimental effect, and so we need to take some cautions when we try to design some clinical trials. So for more information about our sickle cell study, we have a poster today, so you may want to look at the poster session. We also did a study in asthma patients with the platform, metabolomic platform from Nightingale. And in our asthma study, the major risk factor also include age, sex, and obesity and vitamin D deficiency instead of those metabolomic markers. So in our study, we identified decreased level of citrates, ketone bodies, and the 2 amino acids, that is histidine and glutamine. So especially for the decreased histidine, it may be the result of the disease consequence, but not the causative of the disease status. So supplementation of histidine may not be beneficial for the disease. So this need your attention. But for ketone bodies and for citrates, supplementation of these nutrients has been suggested of their beneficial effects, and some clinical trials have been done at this time. And importantly, we identified the markers for lipid metabolism lost significant after we control for the comorbid obesity, because we know that obesity is a major risk of asthma, and obesity is actually cause the dyslipidemia in many patients. So if we control the status of obesity, those lipid disorder lost significant. So again, our results suggest supplementation of some nutrient may be beneficial for the asthma care. So again, for more information about this study, you may want to look at our poster about the asthma and the metabolomic study. So there are some ongoing projects from our side, as well as from our collaborators, for example, led by Drs. Nalini and Dr. Atimadi. So they are working on the metabolomics of obesity and insulin resistance. From our side, we are working more on the metabolomics of some chronic diseases, including blood pressure, diabetes, and other endocrine diseases. So in the future, we are happy to work with more with colleagues in the IHCC consortium. So about how to share our data more efficiently and how we address the security and the regulatory limitations of the data sharing and other resources with you together. So we want to thank all our collaborators and in the IHCC projects, including the Japan NCGG cohort and the East London South Asian cohort, and the Korean Biobank project, and the African populations in the IV-Gen cohort led by Professor Renzi, also the ELSA Brazil project, also the interval project. Especially we want to thank for the platform from Nightingale Health company, but also their experimental assay for our project. So we are happy to take your question and discuss with you about some questions.
Thank you, Huiqi. And maybe just stop sharing, Huiqi, and we can then sort of see if there are any questions from the audience. But I mean, obviously, polygenic risk and genome inferred risk assessment, is are sort of, perhaps an extension of the further, because in the GERA score, you incorporate the polygenic risk, but you add family history, you add certain medical conditions. And in the project with Alzheimer's disease, I mean, ApoE, of course, is a very important risk factor. If you have the E4 haplotype, the risk is certainly substantially greater. So by being able to incorporate that into the polygenic risk is certainly clearly valuable, as you showed. But you also showed that the transgenic risk, if you actually focus on the genetic variants that underlie the traits in diversity populations, you have a very comparable sort of polygenic risk prediction for a particular disease or a phenotype as you have in the European ancestry, and it's based on different markers. So when you combine them together, this could be additive, and there are obviously a number of companies that have sort of started to look at polygenic risk and are offering this as potentially clinical services, which is perhaps a bit too early, but clearly, if done correctly, this is a very valuable approach to assess for risk and identify individuals who perhaps should undergo a certain intervention. For example, as we did in collaboration with the Davos Alzheimer program, where we identified, number of individuals with high polygenic risk score, and these individuals can subsequently be, followed for potential intervention, because they are going to be much more likely to come down with Alzheimer's disease than individuals who have a low or medium type score. So I think we will see polygenic risk really come into play very significantly in the sort of the clinical context in the very, very near future. Question is if there are any questions that have come up. I don't know if in the chat, I don't see any in my chat, but maybe you need to go into the WOA program in order to see it.
Hákon, people can just ask questions if they unmute themselves.
Okay, so they have, yeah. So please feel free to ask any questions, but obviously, what we have demonstrated is sort of the added value of IHCC in the context of generating polygenic risk score that is trans-ethnic, based on trans-ancestry approaches, because no one has been able to do this, and it's very unique given the constitution of the cohorts that we have in IHCC. And not to mention to be able to basically just, put together and train an algorithm and then send that algorithm to multiple different sites. No data needs to get transferred anywhere. People run these algorithms locally, and then we just share results and we meta-analyze the data. This is very, very unique, and certainly not something that I have seen published by anyone. So I think we're going to be the first actually to do so.
Hákon, I have a question. This is Cindy. And I apologize if this was maybe talked about earlier. I popped into the other session and then came over, but I wonder how you envision, or how you might envision, as we advance our understanding of different ancestral backgrounds in genetics, which we're still, improving our representation there. How do we iterate these polygenic risk score models to bring in those real world data, even as we try and move these trans-ancestry polygenic risk scores toward the clinic?
Yeah, I think it's interesting because when we drove the polygenic risk score, we went into the European ancestry publications. We pulled out what was there. We went into any smaller patient population of other ancestries. And when we combined this together and we're able to subsequently go ahead and validate this in a cohort from the eMERGE consortium that we had access to, we actually came up with very comparable polygenic risk across Asia, across Africa, across South America. So I think it's sort of, you are pulling out small effects from these multiple genes involved, and it enables you to do it. So I mean, I think it's going to unfold really in such a way that we are not going to have to do, multi-hundred different polygenic risks, to apply to different populations. I think a trans-ethnic score like this is going to be very robust in addressing that.
That's good news. Thanks.
Hákon, we're out of time, and I want to direct everyone to go to the last closing remarks session. They would have to leave this session and go to the agenda and go to the closing remarks to hear the closing remarks. I will invite everyone to attend tomorrow's session on day 3. It will be mostly dedicated to results and interests in LMICs around the world. So thank everyone for participating in this today.
Thank you very much. I appreciate it.
Thank you.